Can you see it okay? Perfect. Okay.
Perfect.
Yes, I'm going to summarize everything in what we are doing in risk and resilience in the lab, as it was mentioned. So I'm now professor in the psychological and brain sciences. I joined last July. I was actually at Mass General Hospital for several years. And just to give you an overview of what we do in the lab, so I have it here on the left, kind of like have a multimodal approach to understand risk and resilience. And we are using genetics, and I'm going to explain more about that in a minute. We use neuroimaging techniques, including structural and functional MRI and PET imaging, and also going to provide some of the examples. We use blood biomarkers, including amyloid tau and markers of neurodegeneration, inflammation, and so far. Also going to be showing some of that data. And we also use cognitive assessments, including like memory, executive function, and other tools to detect early impairment. So I don't think I have a lot of time for that part, but like I will be happy to answer any questions moving forward. So just to start and make sure that everybody, you probably have seen this many times, but just to want to make sure that everybody is on the same page. So we are talking about dementia. We are thinking about an umbrella term, like the saying is no an equivalent to Alzheimer's disease. Alzheimer's is explained about the 60 or 80% of cases, but it's only one of the possible etiologies of dementia. So in the United States in particular, like Alzheimer's disease explains, I said, the majority of cases. As you can see on the right, about 6.7 million individuals are impacted by the disease. So we are now considering, you probably already know this too, but the same, just to summarize. So we are talking about Alzheimer's disease in different stages. So we know that there is a phase of mild cognitive impairment that precedes dementia, and we also know now that several changes happens in the brain several years before the individual has symptoms in what we call now preclinical Alzheimer's disease. So one of the goals of understanding this preclinical Alzheimer's disease has been for us to see how we could potentially delay the onset of symptoms and in the future, hopefully prevent Alzheimer's disease altogether. We're talking about Alzheimer's disease, so you may already know this too, but just to make sure that you know that what we call sporadic Alzheimer's disease is usually explained by individuals that start having the disease after the age of 65. And there are several risk factors, which age, older age, family history, and APOE4 are some of those important risk factors, especially for late onset sporadic Alzheimer's disease. Then we have another type of Alzheimer's disease that is what we call familial Alzheimer's disease. And in this case in particularly, only a minority of cases are explained by this type of the disease. These diseases are explained by mutations in three genes, like presenilin 1, presenilin 2, and the APP genes. And different from the most common form of Alzheimer's disease, individuals usually develop symptoms before the age of 65. Is they carry a mutation, they're pretty much destined to develop Alzheimer's disease later in life. And depending on the type of mutation that they have, the age of onset will be also determined. So we are thinking about early detection of AD-related changes, and this is the part of preclinical Alzheimer's disease. So one way to study this is to understand biomarkers or identifying these biomarkers of the disease. And biomarkers, this is not unique to Alzheimer's disease, but in general, biomarkers can help us to detect Alzheimer's disease or disease early in the process. It can help us to stage the disease, as well as monitor the progression and the response to treatment where there are treatments available. So in my group, we have been using, as I mentioned, neuroimaging and blood biomarkers to characterize the disease progression in Alzheimer's disease. And in general, there are two ways to go about understanding biomarkers. One is, of course, using the sporadic form of the disease, or in this case, older individuals that are at increased risk to have the disease, or we can use the familial Alzheimer's disease. And as I mentioned, so this is pretty much part of what we do. So I'm originally from Colombia, and in Colombia, we actually have very large extended families with neurodegenerative diseases. We have people with Alzheimer's disease, but we also have individuals with frontotemporal disease, Huntington, CADASIL, that is a type of vascular dementia, Parkinson, and others. And for years, so the group in Colombia, there is a group of neuroscience that was led by Dr. Francisco Lopera for several years, decades. We have been characterizing these families all the way from using clinical assessments to genetic characterization, neuroimaging biomarkers, and now most recently bringing these families to clinical trials. So my main contribution here has been in the neuroimaging biomarkers and moving forward as well. So who are these families? So the families with Alzheimer's disease are the ones that are going to be focused in the presentation today, are individuals that carry a genetic mutation representing one. This is the mutation that we call the PISA mutation, that is the E288 mutation. Individuals with this type of the disease, they usually develop mild cognitive impairment at the age of 44 and dementia at the age of 49. So there are very similar trajectories of this type of disease and what we call sporadic Alzheimer's disease, but of course there are also some differences. Right now we have about 5,000 individuals that belong to this kindred and about 1,500 that we have found like carriers of the mutation. So we were thinking, and this is one of the earliest work that we did to characterize those changes of the biomarkers in these families. So I'm the PI, the principal investigator of what we call now the Colombia-Boston biomarker study of autosomal dominant Alzheimer's disease. And this is, as the name says, a collaboration between Colombia and Boston that initially started at Massachusetts General Hospital. Some of the earlier work that we did showed that when we actually compare carriers to non-carriers using fMRI, and in this case, task-related fMRI, we found that the carriers, the older carriers, the younger carriers, and even the children, they have abnormalities in the way that their brain were functioning. The two things that we highlighted in this work is that they actually have hippocampal hyperactivation and they have less deactivation of parietal regions that, as you may know, so the parietal regions are part of the default mode network, and there was already some abnormalities even in the children in those regions. So we also had characterized the trajectory or the thinning, the cortical thinning in these individuals, and we now know there are two main cut points in the trajectory that around the age of 17 and the age of 32 are the 2 points where the carriers start having greater cortical thinning, and then after the age of 32, they start declining very slowly, but they decline until they have a dementia. And in 2014, we started the project that we were bringing a couple of them. Actually, about 80 people came to Boston from these families to get amyloid and tau pathology. And as part of that work, we were able to characterize the trajectory of amyloid accumulation. So we learned that carriers start accumulating amyloid about the age of 28, like several years before symptom onset, that is 44. And then they also have tau accumulation in the late 30s, around the age of 36 or 30. So the same several years before symptoms. And we also learned that when we compare like the pathologies like amyloid and tau pathology was more closely linked or related to clinical progression than amyloid pathology. So in this paper that we published now a few years ago, we tried to pull this and remember what I showed earlier about the abnormalities in functional connectivity or functional and brain function. And now with some data that we have in amyloid and tau pathology, we put the modalities together and trying to see if there was anything that we could actually predict in terms of brain function based on the pathology of these individuals. And everything that I'm showing right now, just to remind you, is with individuals that are now symptomatic, they are cognitively unimpaired on these preclinical stages of Alzheimer's disease. So we found that even at the very, very incipient levels of tau pathology, the connectivity in the regions that are important for the pulmonary network, they were already disrupted. And those regions, as I was saying, like it was linked to the levels of pathology, including especially in tau pathology, and from very early stages of tau accumulation. So we also characterize the plasma biomarkers, and these are kind of like the two seminal papers that we have. The first one, it was the paper that we published in 2020 when we showed that using NFL, that is a marker or a neurodegeneration, the carriers we were able to distinguish the trajectory carriers versus non-carrier in the early 20s. And the same year or later, we characterized p-tau 217 for the first time in this kindred, and we found that p-tau 217 was a little bit more sensitive than NFL. And in this figure that I'm showing, we pretty much compare head-to-head the two biomarkers. And as you can see, I don't know if you can see my arrow, but pretty much the carriers were distinguished from the non-carriers a much earlier age, and also like through the trajectory using p-tau 217. So with that in mind, the next study that we did, and with the sample that we had from COLBOS, was the same, like I had to say, in this previous work, we have more than 2,000 blood samples from this kindred. But here, because the COLBOS participants, as I was saying, they are just a minority compared to those 2,000, we actually went and looked into the ones that got blood biomarkers at baseline, and then they came to Boston several years later for neuroimaging for amyloid. Yep.
And at least thought some of the.
Sorry. Okay. So while pretty much we had baseline plasma at baseline, and then neuroimaging, tau and amyloid PET about 8 years later. And we were able to find here, let's say the sample size much smaller. As you can see over here, like we pretty much replicated the same associations, which I'm not showing, but we also found the amyloid accumulation as measured by PET, and also tau pathology was linked to baseline levels of plasma p-tau 217. In other words, we found that the baseline tau plasma predicted accumulation or amyloid and tau pathology almost 8 years later, and still, as once again, in individuals that were cognitively unimpaired. So with all of that, we have several projects that we are working on in the lab. This is just to give you a sense of what we have, the same characterizing biomarkers and protective factors for dementia and for cognitive decline in particular, following these families. So everything from sex differences, sleep, physical activity, cognitive biomarkers, stress, and neuropsychiatric symptoms, and many others. And in the next couple of minutes, I'm just going to highlight a few studies that are linked to risk factors and protective factors. So you're probably familiar with this. The reference didn't show up. This is from the Lancet report in 2024, when they actually identified 14 risk factors in the life course that predicted dementia, and they were saying like potentially can modify 45% of cases or dementia. So taking this into account, they're saying we have tried even before the 24 report to characterize these factors in the Colombian kindred. So this is one of the papers that we have. So the first one, that is kind of like the risk factor that is more prominent, sporadic Alzheimer's disease and also other dementias, is the cardiovascular risk factors. So this paper that was published back in 2021 used the Framingham cardiovascular risk profile, which you probably are very familiar with, and studied carriers versus non-carriers to see if we could find any differences. And then we also compared these to white matter biomarkers using MRI. And we found no significant associations. There was no difference between the score, and there were no significant associations between the score and, in this case, white matter measures in the carriers when they were asymptomatic compared to the non-carriers. Further work has shown that this seems to be more relevant by when they actually start developing symptoms and they are already in MCI stage. So linked to that, we also have study physical activity, and this is one paper that we published just now about 2 years ago. In this paper, it was actually conducted at the time of COVID. Most of the data was collected at the time of COVID, which was already a limitation because, as you may remember, we didn't have a lot of opportunities to go around or walk or even pretty much go outside and so far. So I think that was one of the limitations. We didn't find any associations with physical activity here, which was measured using a Fitbit that they have to wear for about 3 weeks, at least 3 weeks. And we linked this pretty much the number of steps in this case to cognitive performance and plasma biomarkers, and we didn't find any association in the carriers versus non-carriers. And it was one of the points to discuss here. As I mentioned, it was most of the data collected at the time of COVID. All the individuals reported very little physical activity. It's the same. We are hoping to continue with this work and maybe in the future repeat some analysis to see if the physical activity can actually predict or cognitive change, like over time, or like when we actually have more vigorous physical activity as well. This work is another important risk factor that is not included in the 14 that the Lancet reported, but is one that we know that is very important for Alzheimer's disease and other dementias, is sleep. So for this study, we just published actually like maybe 2 months ago. And we found what we did. Let me just explain briefly what we actually did for this study. So the participants came to the clinic in the evening. So they were tested before they went to bed, and they actually stayed in the hospital and the clinic for an overnight sleep. So we track PSG activity, polysomnography activity during the night, and the day after they had breakfast and everything. So we actually asked them to remember some of the cognitive testing that was done the night before. And then we also called them again 1 week later, 7 days later, to see if they actually remember any of those tests that were given on the night before. So interestingly, what we found is like when we look at the activity in the PSG, we found that the carriers versus the non-carriers, they have like less parallel spindles, like sleep spindles, as compared to the non-carriers. So that was the first thing. So they had reduced parallel sleep spindle, and then that activity, that reduced activity in this case, was linked to a lower memory recall, like 7 days after, even if we didn't see any differences in the day they were tested, 7 days later. The carriers had lower performance, of course, and that was associated with the reduced parallel sleep spindle power.
I can't understand.
Okay, so the other factor that we have studied is depression. And in this paper that was published also in 2023, we pretty much just asked individuals to complete the GDS, that is the geriatric depressive scale, and compare the baseline levels of depression with hippocampal volume changes over time. And we found that the same, like the lower hippocampal volume was associated with depressive symptoms in the carriers, but not in the non-carriers. So this is what I was talking just a minute ago in terms of like what factors can be modifying. One of the early factors that are linked here is education. So for education, so we wanted to explore if education have a role in the trajectory and the onset of symptoms. And one of the major genetic risk factors, as I mentioned, was ApoE. So we also wanted to see if ApoE, including like ApoE4, ApoE2, and of course ApoE3, if they have any role in the onset of the disease. So what we found, and this is the same going back to the larger sample, like almost 2,000 individuals, we found that the APOE4 was associated with a accelerated onset or age-related cognitive decline. They usually develop symptoms at the age of 44, and it's pretty much that was the critical point or differences between the carriers and the non-carriers. And for the APOE2, that is usually associated with protection. So we found that the carriers of the presenilin 1 that also carry the APOE2, they have a delay onset or cognitive decline. And the same about the age of 41 is when we actually distinguish those two groups. We also observed that APOE4 had an impact in the onset or MCI and the onset or dementia, considering like the ones that actually carry APOE4, they have an earlier onset or those conditions, MCI and dementia diagnosis. And listen, I didn't show it, but also we found something similar with APOE2 in terms of having a delayed age for diagnosis. We look at all the different factors that we have access in terms of like socio-demographic factors and socio-cultural factors, and education was the only one that was actually linked to the trajectories or clinical onset. And here we found that education was actually protective and was able to moderate the effects of APOE genotype. What this means is that pretty much like the ones that actually carry the two mutations, like they carry the autosomal dominant mutation and they also carry the APOE4, the APOE4 variant. So if they have higher levels of education, they have a slower decline compared to those that actually have lower education. So education was actually able to protect even at the highest risk or genetic risk in this case. This is pretty much what I was just saying, highlighting the role of, first of all, the APOE plays a role modifying the age or clinical onset or in presenilin 1 carriers, and also that education has a protective effect in this impairment. So in 2016, Dr. Francisco Lopera and I started a project that we call it a COLBOS Extreme. And it was as part of this collaboration between the group in Colombia and the group here that we were going to study those that pretty much didn't follow the same typical trajectory of the disease. So we started looking at factors of protection, including like resistance to pathology or cognitive resilience, even in the context of brain pathology. And the first case that we were able to identify was the case of Doña Liria Piedrahita. Doña Liria, she was a member of the family, meaning she was a carrier of the presenilin 1 E280 mutation. She was expected to develop MCI at the age of 44 and dementia before the 50s, but she only showed memory symptoms in her 70s, and she was diagnosed with MCI at the age of 72, which is almost 30 years after the expected age in this kindred. She was relatively healthy, and the only thing that she had was that she had high levels of cholesterol, but nothing else was there. So she came to Boston as part of the project. Actually, she came twice, and we were able to see what was happening in her brain in terms of pathology. We found, as you can see here in the figure, that she had really high levels of amyloid pathology. Everything that is red here is the amyloid pathology, but she had very limited levels of tau. As you can see here, like almost no extensive tau pathology compared to somebody that is typical in these families at the age of 44. The age of 44. I'm not showing it here, but we also found that she had very limited neurodegeneration, brain atrophy, very limited as well, and also in blood biomarkers. We ran in collaboration with some colleagues at the Massayaneer some genetic analysis, and we found that she had two copies of the rare mutation, that is the Christchurch mutation. And since then, the same, like we have learned a lot about the Christchurch mutation, and here I put like some of the main findings, and the findings keep coming. So the Lancet Neurology actually published a review that we wrote last year, which now seems a little bit outdated. But as part of the work that we reported, so the first thing that we learned from Christchurch is what we proposed in the original paper, that is the ApoE3 Christchurch shows reduced binding to the heparin sulfate proteoglycans, the HSPGs, and that has an impact in the tau accumulation in AD-affected regions, particularly in the medial temporal lobe and the entorhinal cortex. This is, as I said, something that I was first proposed in this paper. We also hypothesized here that this may lead to reduced neurodegeneration. Since then, there's like some of the papers that I want to highlight here. It's like Dr. Nelson, actually, in 2023, she published the paper using ISPSS and mice studies, and they showed that Christchurch also have a protected effect against ApoE4, which, as you may remember or you may know, so is the main genetic risk factor for sporadic Alzheimer's disease. They showed that those that actually had two copies, they have a reduced tau pathology, neurodegeneration, and neuroinflammation. And those that only carry one copy, they have a partial protection, including reduced neurodegeneration and neuroinflammation. They didn't see protection against tau pathology in those that only had one copy of the disease. In the original paper, based on the mechanisms that we suggested, that it was the binding with HSPGs, so we proposed we developed an antibody that is the 7C11. This is work that Dr. Claudia Marino has led. In 2024, she reported some of the advancement with this antibody, showing that the antibody actually is able to mimic the function of ApoE3 Christchurch, and also it showed reduced tau phosphorylation in the ApoE4 mice as well, and decreased tau pathology in a mice that actually carry the MAPT mutation. So there is more animal work that has been published. This is some work from Dr. David Hochman, when they actually showed that the mice that actually carry the Christchurch, they had reduced amyloid pathology, and they also had amyloid-induced tauopathy, which also leads to reduced neurodegeneration. They also reported some improved microglial response, and in the paper that was actually published last year, they also found that there was a protection against tau-induced synaptic loss, myelin loss, and reduced reduction in hippocampal theta and gamma power. And the same, this is based on what we reported. You want to learn more, you can start with the review, but since then there are already some papers that are also support the protection against neuroinflammation, and particularly and tau pathology as well. So in our case, working with the families in Colombia, we went back to the, like almost, as I mentioned, 2,000 samples that we have, and we genotyped those samples to see if we were able to identify more individuals with Christchurch. And we actually reported this back in 2024 at the New England Journal of Medicine, when everything that is green here are the carriers or the Christchurch. We actually were able to identify 121 members of the family that carry one copy of the mutation, and including 27 that carry the two mutations, and the PISA mutation and the Christchurch. We were able to track the clinical data, most of the sites, of course, doing it retrospectively with all the data that was available in the group. And the carriers of only one copy, they had approximately 5 years delayed in the onset or MCI. And they also have an approximately 4-year delay in the onset for dementia. So we have two people that actually went through the process of getting PET imaging. One of them came to Boston for amyloid and tau, and then the other one actually have FDG PET in Colombia. And here I'm showing the results on the right, or the one that carry one copy, or the Christchurch. And interestingly, this is only one individual, but we found that these individuals have very similar amyloid pathology compared to the typical carriers, but the levels of tau pathology, as you can see here, is very limited, especially in middle temporal lobe regions. So with the individual that actually came for FDG PET, we also found that they have preserved glucose metabolism, which was something that we also found in the homozygotes. So now we are following these individuals, and from the 121 and 27, of course, like many of them, because we collected this data like for the past 30 years or more, many of them have died. So others are very young because we are only characterizing. So we are trying to collect biomarker data, longitudinal biomarker data in as many as we can. And starting with the same process of characterizing the structure, the functional, the metabolism, and the pathology of these individuals to see how is that in Christchurch impacting their risk or their protection for Alzheimer's disease and dementia. So this is ongoing work, but I just want to show an example that is we have about 40 individuals that actually went through MRI and FDG PET that are carriers of only one copy or the Christchurch. And we are comparing here from the non-carriers. And interestingly, the same we are seeing that the carriers, the Christchurch carriers, they have a different pattern or structure, function, and even a glucose metabolism. And the data, as I was saying, this is still work that is ongoing, but it seems that this difference seems to explain some of the protective phenotype, including the better cognitive performance that links to like better working memory and better episodic memory as well. So more to go, but I just wanted to share what we were doing with that project as well. So design, so like after Christchurch, and because of time, I'm not going to be talking about all the other cases, but the second case, at least I can briefly mention. After Doña Lidia, we also identify a second case, that is the case of that we found the mutation of reelling COLBOS, explaining the disease. This was also an individual that only developed symptoms in the late 60s. And the same, he also was able to came to Boston for neuroimaging. And different from Doña Lidia, we found that the second individual have amyloid and tau, very similar to the typical carriers, but there was a region of the brain that was protected, that it was entorhinal cortex that didn't have a tau pathology. Some of the molecular analysis identified this reelling COLBOS also having an important role in the binding of HSPGs, and that's something that is now like also under study. We are now characterizing the third individual that has been identified that is protected, and yeah, hopefully soon, the next few months, I will be able to share more about that one too. And similar to what we did with the Christchurch, we're also characterizing the individuals that carry the reelling COLBOS to better understand their protection. So hopefully with all of this, so you get a better idea of how much we can learn from the families with autosomal dominant mutations, especially when we're trying to understand disease progression, trying to find better ways to detect the disease, and of course, trying to identify protective factors and targets for therapy development in the future, and of course, clinical trials. The same, the latest work, and I think that one of the most exciting things that we learn from these families is that there are genetic variants that can be protected even in this context, including the Christchurch mutation, and the Christchurch may delay or may be protected against cognitive impairment and likely linked to reduce tau pathology and lower neuroinflammation. And the same, this is kind of like pointing to a very promising pathway for resilience-based therapies, and hopefully in the future, we will learn more and more about how to move these therapies to clinical trials. So future studies are needed, of course, as part of the, as I mentioned with COLBOS, it's now a longitudinal study. So yeah, in the future, we will learn more about the trajectories of these biomarkers over time, and also a link to cognitive changes. And of course, there is more to learn from the escapees. As I was saying, we are working in the third protective mutation that it seems to be linked to protection. And there are more individuals that we will characterize in the future. So just to conclude, I wanted just to mention some of the ongoing work. So now we have the PRADA study, that is a protection against dementia in Antioquia, following a similar path, as I was saying before, starting with the patients and characterizing the genomics, using basic science. And so far, we are extending this work or identifying potential protective individuals to other types of dementia disease, but we also extended it to frontotemporal dementia and CADASI, like the vascular dementia. And we hope to see if we can identify those potential mutations. It looks very promising so far. And for those that are interested as well, so of course I didn't talk about this work today, but and also the PI or the Boston Latino Aging Study, that is the study of biomarkers we are doing in individuals older than 55, including Spanish-speaking Latinos and Portuguese, Brazilian speakers as well. And the idea is to see if we can identify those risks and protective factors in these individuals. With that, I would like to thank all the collaborators, people in my lab, and especially the families from Colombia for their commitment to research. And I also wanted to take the opportunity to invite you to a symposium that we are organizing in September here in the Charles River campus. In case that you want to learn more, we'll have a dedicated feature research session on protective variants that we have identified in Colombia. And with that, thank you very much.
Awesome. Thank you so much. And feel free if you want to send me that as well. I can send it out to everyone about the event. I think that would be great. So I'll open up the floor to anyone who has questions. So take it away. Yeah, feel free to unmute.
I have a question, Ben Walzen. Of course, you've done such amazing work. I have a strong interest in tau and neurodegeneration and mechanisms of neural degeneration. What's your sense on how, how it, what the field has found about how the Christchurch mutation is kind of limits tau pathology?
And it's an idea that we are still learning, but something that we, for instance, in the first line of research that I described, we actually are identifying that this binding between ApoE and, in this case, HSPGs play a role in the tau pathology and how tau pathology actually propagates. So that's kind of like the main finding of one of the studies.
So you think it's preventing the propagation rather than the formation in the cells?
Exactly.
Interesting. Thanks.
Exactly.
Well, I guess I'll jump in. Hi, Yakeel. I'm Lindsay Farrer. I really enjoyed your presentation. It brings back a lot of memories from the time I was working with Ken Kosick in this project many decades ago. So I have a couple of questions. The first is related to the Christchurch So it's very clear from all these studies that the mutation has impact, AD-related impact. But is anything really understood about how this mutation affects the ApoE protein? What characteristics does the protein have that are different from those who don't have this mutation, especially since it's on an ApoE3 background?
Yeah, and that's like one of the first thing that I was reported with the mutation, like the ApoE3 Christchurch mutation, and that's why it has the name of Christchurch, because of the city of Christchurch. It was actually linked to a lipidomics and a lipid, like how it impacts the lipid activity. And like as I mentioned in the original case, she had a high levels of cholesterol. That was something that was linked there. But there are some studies, like one of them actually from our collaborators, that are showing that the lipidomics are actually abnormal or altered as part of this interaction with the ApoE and the Christchurch. So definitely something there that we are learning more and design, like also the link between the lipidomics and how that impacts and translates to tau pathology and neurodegeneration as well.
All right, if I can follow up. So you were very fortunate to identify what ostensibly is a protective factor, the Christchurch mutation. But as you showed, it's in homozygous form in only a small portion of this very large kindred. And there's probably a lot of variability in onset among people who don't have it. And so my question relates to a more general one that those of us working in the AD genetics field are wondering, have you or has your group embarked on any project to identify other genetic factors? And if so, how are you going about doing that?
You mean like identifying other factors in?
Yes, so a genome scan.
Exactly. So yeah, as part of that, as I mentioned, so we have been able to identify other potential protective mutations, but they are still in the study in individuals that had also the clinical onset at much later age. So it's kind of like tracking the kindred. But related to what you were saying before, not just in the kindred, because the saying is one thing is that what we can see in the kindred, but I just 2 weeks ago I actually submitted like the request on the Framingham study after you guys came here to present, because I wanted to explore even in the data that we have in Framingham how common is the mutation, if it is at least one individual that have the mutation. We did something similar to with the UK Bank. So we actually requested the data for the UK Bank. There is one publication that they had that 20-something individuals from the entire UK Bank actually had the Christchurch mutation, like one copy of the mutation. But in the UK Bank data, they found that in these individuals, even if there were only a few of them, they have better working memory compared to those that didn't have the mutation. And with the data that I briefly showed that is ongoing data collection for us, we are replicating and seeing something similar related to working memory. So that's kind of like just extending it to other group of people that are beyond the kindred. But with the kindred, yeah, with the kindred alone, we are exploring other variants as well.
Okay, so my question wasn't as much emphasizing finding this mutation elsewhere, but looking for other genetic factors, even within this kindred,
Yeah.
which I suspect there must be. Has anything been done along those lines?
Yes, exactly. Like one thing is like the same, like Ken Kosi continues to do analysis at the large level for the genetics. They actually have a publication, I believe, 2 years ago. The first author was Juliana Acosta, when they actually tracked and reported like some of the variants in the whole sample, the genetic variants that could actually be linked to like increased rates or decreased rates. Like that's one paper in that. And they also extended to not just Colombia, but also other countries in Latin America. So like standing that at that level of large analysis. The analysis that we, like my group in particular, is doing is more like a small scale. So pretty much we're actually starting, as I mentioned, starting with the clinical information, looking for those individuals that are protected, and then we do a whole genome in those cases instead of the large kindred.
Yeah, I'm not sure whether this approach has been taken, but you've got the perfect sample of people to apply what people now don't even attempt to do anymore, and that's linkage analysis. That is, in a kindred like this, it's much more efficient than simply applying a case control strategy. And if it's something that you're interested in,
Yeah.
my group would be more than happy to assist you with that.
I would love that. I will follow up with you on that. Thank you. Yeah, that would be great.
All right, I'm going to shut up and let other people ask.
Thanks so much. Does anyone have any other questions? Oh, there's a question in the chat. I can read out loud. Thanks for a great talk. Do you think the protective variants you found could signal towards a common mechanism for tau pathology or possibly point to different mechanisms? For example, I think the RELN COLBOS variant also affects the relationship with HSPGs. Would love to hear your thoughts.
Yes, I briefly, I apologize because I actually thought that I have the information on the railing COLBOS there, but I probably missed it. So as I briefly mentioned, like one of the points on connection or what we discussed, I think that it was Irene. Yeah, Irene. In the Lancet Neurology, we review that I mentioned. We also discuss and propose a common pathway that we call it like a resistance pathway, because there are several elements that are common between the Christchurch and the Rilling colbos. So, and even like the same that has been discussed in other mutations, like the Jacksonville, for instance, at some point. So those commonalities, we kind of like put it together and discuss it in the paper. But the one that we started highlighting is that one of the ApoE and the HSPGs and how that pathway can be relevant for resilience.
Awesome. Thank you for that. If anyone else has any questions, we still have a few minutes. Feel free to put those in the chat or you can raise your virtual hand. I think we have another one in the chat. Great talk. Is the sparing of the entorhinal cortex observed in the second ApoE-CH patient a common feature among the carriers with delayed onset of dementia?
So the short answer is yes. So the difference is that so in Christchurch, the protection, for instance, against tau pathology extends to the entorhinal cortex. It's not just the entorhinal cortex. It's actually extended to the hippocampus, the inferior temporal, and other regions. The Rilling colbos, that is the second case that I mentioned, it was very limited and restricted to the entorhinal cortex. But the entorhinal cortex, or the protection of the entorhinal cortex, is common in between the two cases.
Hello. Great talk, great work that you're doing. I have a question that's not directly related to your research, but I'm just wondering if you've had observations. With this group, have they have members been using some of the amyloid clearing drugs like lecanemab and things like that? And if so, have they been reacting well? Have there been any problems? What has been the experience?
So we actually got a funding from the NIH to run the clinical trial using donanemab. We haven't started. So ask me again in a year or two and see how it goes. But we have now like they finally get all the approvals to start enrolling, and hopefully we will be enrolling the next few months. But that's the first one. So nothing has been done before with those treatments. So the first clinical trial that we conducted several years ago, that ended 2 or 3 years ago, was with crenezumab, and that was negative. So it didn't actually modify disease onset.
Interesting. Interesting to hear the results. Thanks.
Awesome. Well, if anyone has any other questions, this was great interaction. Love that everyone had such awesome questions and we're able to have this time to do that. But yeah, if anyone doesn't have any other questions, we can end a couple minutes early. But thank you so much. And I'll be sure to send out anything that you send to me for everyone. And I'll be posting this recording onto the website for anyone to review afterwards if they'd like.
Thank you.
Awesome. Thank you so much, everyone.